So this is your video, there we go, on proteins. It does go with your guided notes, so please make sure you have those out at the same time as we go through this. We're kind of continuing on in biomolecules, but we're keeping the same themes we've been addressing this whole time. And that is this idea of looking at these organic molecules. So these ones that cross all living things are so important we can find them in any living thing. What are they? And then how is their structure and function related? So clearly, these proteins, these molecules, carbohydrates, lipids, things we've been studying are hugely important, which is why we find them in everything. They're doing some job that's essential. And we're going to kind of appreciate sort of what is that job? So, what needs to be done in every living thing? And how does their structure help them with that super essential job? When it comes to proteins, we know we can look at kind of the elemental composition as a way to differentiate between our four categories of organic molecules. So we're going to add our last kind of elemental composition here. So proteins are made up of carbon, hydrogen, oxygen, and nitrogen. We have the full proper name for each of those elements written out underneath, and then also the single letter shorthand. These ones do each go by one letter, not the two. And you just want to make sure that if you see a capital letter C, that you also know that that's referring to the element carbon. They also, like our other categories of organic molecules, are similar in their structure in the sense that there is a small unit that you basically repeat and connect to itself over and over again, almost like stacking Legos to make a tower in order to make your large molecules or your polymers. The name for those small units when it comes to proteins are called amino acids. So that is the name for their building block. And we're going to look at their structure in about a second. One of the things I wanted to stop here and kind of recognize is if you are thinking back, what did I learn in terms of elements for for carbohydrates, for lipids, and for nucleic acids, is you should realize that we've hit on 5 of the 6 most common elements. But there's one we haven't quite addressed yet. And I'm curious if you can guess which one that is. So maybe you are in your head right now and be able to be like, yeah, you know what? She's right. We talked about carbon, hydrogen, oxygen, nitrogen, and phosphorus. But what about sulfur? And don't worry, I'm going to tell you guys what's up with sulfur in about 2 seconds. But in order to appreciate kind of where sulfur pops in, we'll talk a little bit about the structure of our building blocks of our protein, so the amino acid itself. This goes with questions 3 and 4 in your notes. So question 3 asks you to identify the functional groups. And we'll talk about those in a second. And also to sketch. So number 4 asks you to include a diagram of an amino acid. You'll notice that over on the left-hand side of my screen, I do have that basic diagram. So you guys would be copying this down. Do include the labels for the different groups in here as well. What functional groups are is they're little clusters of elements that when we see them kind of in this particular arrangement on different molecules, we can predict something about their behavior. And we notice that we have 2 of these said functional groups on our amino acid. We have what's known as an amino group. And so that's on the far left-hand side of my amino acid. It is a nitrogen with 2 hydrogens coming off of it. So that's what this N represents an element of nitrogen. This line connecting it to that element of hydrogen is meant to represent a covalent bond. So remember, covalent bonds are stronger kind of typically of our bonds compared to ionic. So this is going to be kind of a fairly solid molecule here. On the other side, so the right-hand side, I always think of an amino acid as kind of like being a giant T, where you have this central carbon, and then it's kind of like what comes off it, north, south, east, and west. So we just kind of talked about the amino group, which was shown in this diagram as being on the west-hand side. But if we come off the other way, we notice we get our other functional group, which is our carboxyl group. So this is C, double bonded to an oxygen, and then with an OH group coming off the other side as well. Notice that in this diagram, these sit opposite each other, one on the west, one on the east. But you could technically invert these. So put carboxyl on the west, put amino on the east, and it would still be an accurate diagram of an amino acid. The only thing is you couldn't put them next to each other. So that means I couldn't have my carboxyl group coming off the north side of this carbon right here and my amino group right next to it. That would be an inaccurate diagram. They always sit opposite each other because they're rather large functional groups. And so just to kind of make space for the number of atoms that we need in here, they're always going to be across from one another. I notice that I always have a hydrogen. So that is going to just sit up there. And then on the other side, we have this thing called a side chain, and we see letter R. And we might, come from our life of looking at a periodic table, we might be like, you know what? I don't ever remember seeing an element R. And you are correct. There is no element R. What R is used in chemistry almost like X is used in algebra. It represents a variable or an unknown. So in this case, we are looking at an R group to represent something that can be different. Because when it comes to our amino acids, there's part of this molecule that is stable and going to be a constant, and there's another part of this molecule designated by where this R is that changes. And what we find is that in nature, we actually have 20 different variants for what can sit in that R position on an amino acid. And this gives us kind of our alphabet, so our base for creating our different proteins, because we have these 20 different building blocks, these 20 different monomers that exist in the protein language. And these variable or side chain groups, R groups, are going to determine how an amino acid behaves. So this is kind of a general diagram right here, but you'll notice that we have some nonpolar ones. So for example, valine, which is diagrammed right here, kind of where my mouse is. That one is going to mean that it is not going to try and react with other things. It's going to be like a lipid in the sense that it's not interested in interacting with anything else with a charge. Whereas down here, we have serine. So this is a polar one, which means it is going to be interested in interacting with other things with charge. And these unique sort of chemical properties of each of these different R groups is what's going to contribute to what we'll later see as the bending and folding and the creation of the final shape of a protein. The sequence that these letters end up in, the sequence that these amino acids end up in, is going to determine where that folding, bending, and interactions will occur based on the chemical properties of these R groups. Now, it asks you guys to talk a little bit about kind of how we get such massive diversity in our proteins. So this one's a little bit of a longer rip, but I think it'll kind of connect some big ideas for you guys. So amino acids, because we have that carboxyl group, we've got the amino group, and then we just have that variable side chain, they all have the same basic structure. So what happens is when you have amino acids forming bonds between one another, it's actually the amino group on one, so the nitrogen-containing functional group, that connects to the carboxyl group on another. And because these two functional groups are the same on every single amino acid, they can all bond interchangeably. It doesn't matter which of your 20 amino acids we're looking at, you can bond any one of them to any other one. And then, because we have this rule, hey, they're all interchangeable, by varying length. So maybe I have a protein that has 20 amino acids, and maybe I have a protein that has 100 amino acids. And by varying the sequence, so which order these 20 sit in, you get a massive diversity. And we'll kind of take a minute to look at the interchangeability, and then we'll talk a little bit about what type of diversity. But you can get a huge amount of protein diversity. Pause here if you need to make sure you get this down in your notes. So the analogy I always like to make between your 20 amino acids that I think really drives this point home about how this, again, our big idea here, structure is related to function. So we have this variable group that gives us 20 amino acids. How does that contribute to a protein's hugely wide variable functions? Well, if you kind of think of these amino acids as being like letters in the English alphabet, we have 26 letters in the English alphabet. There are 20 amino acids in kind of the protein alphabet. But we know that in the English language, we can take these 26 letters, and by just putting them in variable length groups and changing the sequence of them, I can get the complete works of William Shakespeare, I can get the Gettysburg Address, I can get the complete works of Dr. Seuss, I can get the Declaration of Independence, I can get all of Steinbeck's work. So I basically can get a massively different collection of information, but I'm using the same 26 letters. I'm just combining them in different lengths and in different orders. And this is exactly what's happening in the natural world as well. We are essentially taking all living things, not just humans, all living things are taking these same 20 amino acids, and they're just rearranging them. So they are creating them in different lengths, different sequences, and they are making different proteins. Now, we refer to the different proteins an organism can make as the proteome. And the cool thing is I was doing some research on this because I wanted to make sure that my estimates were good, but this is actually an area that research is currently at. So we're kind of in modern times in terms of what researchers are looking at. And that is trying to figure out, well, how many proteins are in the human proteome? So how many proteins can we actually make? And the current estimate is hovering right around 19,500, but again, ongoing work. So with these 20 amino acids, we're currently at just under 20,000 for novel or new different proteins that we as humans can make just from these. And again, that's only humans. There are many, many, many other animals that are making their proteins as well. So the big idea, in case we haven't gotten it yet, so we have but and we have tub. They have the same letters, but we've rearranged them and completely changed their meaning. We can also have this happen with amino acids. If a mutation occurs in the DNA is sort of the blueprint for what we use to actually determine what sequence these amino acids need to be in. So if there is a change in that DNA or an issue that occurs when that protein is being put together, so we get the amino acids in the wrong order, we end up with a very different protein. So it's like almost swapping letters in a word as well. Like you're kind of like, this doesn't make any sense here. Your body does the same thing. If we start messing with those amino acid sequence, they get a protein and they're like, what is this? How am I supposed to use this? So that is why mutations can be a big consequence. Moving on to the polymer. So the polymer of your proteins are called polypeptides. Poly is a nice prefix. We're familiar with it from looking at carbohydrates with polysaccharides, but that peptide word is a little funky. Peptide actually refers to the specific name for the covalent bond that forms between amino acids. So I told you guys that kind of the functional group, so the amino functional group and the carboxyl functional group on different amino acids connect to kind of form our big chain of our polymer. And so the bond that forms between those two is just called a peptide bond. It's the proper name for it. So when you have this polymer term, polypeptide, it's named for those multiple bonds that form between amino acids to create these large protein molecules. The two things that determine a protein's function is a sequence of amino acids. I alluded to this when we were talking about the idea of butt versus tub. You just swap two letters, but it means something totally different. Same thing with proteins. So the sequence of amino acids is hugely is going to really determine its functionality. And then also the shape of the polypeptide. And 1 and 2 are really interrelated things. I told you guys basically that when we looked at amino acids, that each of these amino acids has their own chemical properties. And those chemical properties are going to determine how they interact with neighboring amino acids. So where are we going to get folding? Where are we going to get bending? But as I swap up these amino acids, obviously those interactions are going to change and I might get a different shape. What we're going to do now is kind of the message moving forward is really going to be looking at and making connections between this idea of amino acid sequence and shape. And so what we want to do is kind of look at what are the stages when a protein is made in terms of the transition it goes through in shape. This is going to help you when we get into mutations in protein synthesis, and also when you start looking at genetics to understand how alterations in this can create problems kind of on the end of shape. So when proteins are first created, they're created at a structure in your cell known as the ribosome. Ribosome is found in all cells because this job of making proteins is absolutely essential. You cannot have a living thing that does not make its own proteins. We have yet to find a form of life where that's true. So we find ribosomes in everything. And when these amino acids are first being strung together, they end up almost looking like beads on a Mardi Gras chain. So there's just going to be this long chain, and we call that its primary structure. Now, just to make sure we're not confused in terms of what's going here, you should be all the way on the bottom of your notes, and you'll notice that you have a chart with 3 rows in it. The top row is where you're going to list what structure. So you would write on that top row, all the way on your left-hand side, first column, primary structure. And you'll notice I did not make you do any fun artistry here. I already gave you a picture of the primary structure of a protein. And then you have that blank row underneath, and you're like, well, what goes here? What I'm interested in you guys keeping track of, what are the bonds or interactions that give you this structure? So if we look at my notes up here, primary structure gives you that Mardi Gras bean chain. And what we have for bonds or interactions is it's actually just the peptide bonds. So the second bullet right here, you would want to summarize this in that last bullet and kind of keep this moving forward. So top row, you're listing what is the structure. Middle, you've already got a nice picture from me. And on the bottom, you're telling me what bonds or interactions actually give you this structure. So it's the interactions between just the monomers. When the ribosome is actually assembling amino acids, it literally is creating those peptide bonds. And again, you have a whole unit to learn about more about this. But you just want to know that you're going to end up with this Mardi Gras bead shape with just the peptide bonds dictating sort of what sequence that chain is in. Now we know that as soon as this chain gets released, we know that amino acids have their own chemical properties. Some of them are going to have positive charges. Some of them are going to have negative charges. And we know opposite charges attract. If you happen to have amino acids with those opposite charges near each other, well, they're just naturally going to start to gravitate and pull towards one another. And this is what we're going to end up with for secondary structure. Secondary structure has two cool forms. There's what's known as the alpha helix. So sometimes in an attempt for those positive and negative charges to get closer to each other and form these weak hydrogen bonds, it causes the molecule to actually curl. So it looks almost like a spirally pasta or a twisted staircase. A little DNA-esque here, although it's not DNA, different molecule. And then we also have another one that's a beta, which is known as a pleated sheet. So this is almost like, I don't know if you guys have ever seen like the crimped 80s hair where it goes like up and down, but that's kind of what a beta pleated sheet is going to look like. But again, for that last row that we're filling in, it's the hydrogen bonds forming between those opposite charges that causes this curling or crimping. In your tertiary, which is a very fun word to say, this is where you have other bonds that are going to come in. So this is not just slight positives and slight negatives being attracted to one another. These are full bonds that are going to form. So we're talking here about covalent bonds, ionic bonds. You also can get what's known as a disulfide bond. So this is where two sulfurs form a connection between one another, which is a very, very, very stable connection that sulfur likes to make. And you will get even more bending and twisting. Now, whereas with the primary structure, I was like, hey, it's just a Mardi Gras bead. And with your beta, where I could say, hey, it curls or it crimps, tertiary structure is going to be hugely variable in terms of how this bending or shifting occurs because it depends entirely on where the amino acids are that are forming these bonds. So this is where you're going to get truly like a curled back, hugely variable structure on itself. Now, some proteins are done here. So some proteins will just make a chain at the ribosome. They might have some curling or crimping that occurs due to charge, and then they might have some additional stronger bonds that form, cause it to fold and bend back on itself a little bit more. Other proteins need to combine with more polypeptide chains. So the example that we have here, a really common one that you might be familiar with, is this is actually a picture of hemoglobin. Hemoglobin is the protein inside of your blood that actually transports oxygen. It has an iron ion right here that's shown in kind of in this gray sort of looks almost like a rice or something right there. And this iron ion gets folded into protein chains. But hemoglobin on its own is not a single protein chain. It's actually 4 chains combined. And again, some proteins are done at tertiary structure. They do not need to combine with others. And other proteins do need to combine with other chains in order to form their functional kind of final form. So that's not all proteins do this. All right, guys. So I'm sure you're probably done here. Go back through, make sure, rewind this video, rewatch anything you need to. You should have everything in your notes on proteins. And again, you'll do more practice with this.